Okay, you guys, in this video, we're going to look at topic 2 of the Unit 3.3 notes. This is when we're going to now start diving into the details of photosynthesis, starting with the first part of photosynthesis, which is the light reactions. And so for the light reactions, all of this is going to take place in the thylakoids, like we talked about in topic 1. In the thylakoids, these are these membrane discs that are found stacked on top of each other in the chloroplast. Just to remind you in this picture right here, these guys right here, these thylakoids, these membranes, the system of membranes that forms these discs stacked on top of each other. This is where the light reaction is taking place. So this is where we're at. Now in this membrane, the thylakoid membrane, so this membrane that makes these discs, you're going to see a lot of different proteins embedded in that membrane. And one type of protein that's embedded in that membrane are these protein complexes called photosystems. So these giant protein complexes here, these are located in the thylakoid membrane, just like this picture is showing you here. And in a photosystem, what this is a special protein complex that's going to be able to absorb energy from sunlight that's hitting the plant, and then take that energy and convert it into chemical energy that's now exist in the form of excited electrons that are going to now travel somewhere and make it to different chemical components. And so if you look here, there's in a photosystem, this special protein complex here involved in photosynthesis, there's a bunch of these pigment molecules embedded in it. So these green little circles here represent pigment molecules like chlorophyll and other types of pigment molecules like carotenoids that are found inside plants. There's a bunch of different pigments embedded in this giant protein complex that absorb energy from light. And then what they do is all these pigment molecules is they take all this energy from light that they're absorbing and they focus it in on this these central pigment molecules, which are a special set of chlorophyll A molecules, so some special set of chlorophyll A pigments right here, where all this light energy is going to be focused into. So all this light energy being absorbed by all these pigment molecules in the protein, they're going to take all that energy and focus it to these central pigment molecules here. And that energy is going to be used to excite a pair of electrons. So there's going to be a pair of electrons here that are going to take all that energy from the light and become really excited. So they're going to turn into high energy electrons. And once they become high energy electrons, they are going to basically be kicked out of the photosystem. They're going to be then moved out of the photosystem somewhere else. And that's what we're going to look at here in topic 2. But that's what a photosystem does. A photosystem is just this protein embedded in the thylakoid membrane that has all these pigment molecules in it that are taking all this light energy and focusing it on these central pigment molecules that are going to then use all that light energy to excite pairs of electrons that then get kicked out of the photosystem, as you guys will see in a second here. And so here's an overview of what's going to happen in the light reactions. We're going to take energy from the light, and we're going to use that energy to make two things mainly. We're going to make ATP and some NADPH molecules. And so what's going to happen overall in this process is we're going to see a movement of electrons, and those electrons are going to start from water molecules. Water is going to be the electron donor. This is where the electrons will start. We're going to take pairs of electrons from water, and then that pair of electrons is then going to get excited and then go through a bunch of different proteins, which we'll see in a second. And then those electrons will ultimately end up with NADP+. NADP+ is an electron carrier molecule involved in photosynthesis who's going to take those electrons that originally came from water and then become NADPH and then take those electrons somewhere else. And so there's different components here that make up the light reactions that we're going to look at real quick, starting with photosystem 2, and then there's the electron transport chain, and then photosystem 1, and then NADP+ reductase, and then ATP synthase. All these guys are involved in the light reaction. So let's take a look at these. Let's start with photosystem 2, and we'll kind of put these pieces together here. So in photosystem 2, let me just show you this picture here. So here's photosystem 2. And like I showed you in a photosystem, you have all these pigment molecules that are going to absorb all this light energy, and they're going to focus all that light energy into a central pigment molecule. In photosystem 2, the central pigment molecules are called P680. It's called 680 is because that's the wavelength of light that it's really good at absorbing. It doesn't really matter. And so, but basically all this energy is going to be focused on that central pigment molecule, and all that energy is going to be used to excite a pair of electrons. And those electrons are going to become really excited because they absorbed all this energy from light, and then they're going to be kicked out of the photosystem. That's what's happening here in photosystem 2. Now, what we have to do then is we have to constantly replace those electrons, those pairs of electrons that are becoming excited and leaving photosystem 2. There's got to be someone replacing those electrons so that we can continually excite new electrons that can then leave the photosystem. And that's where water comes in. So water here is going to drop off a pair of low energy electrons. Water has these low energy electrons that it's going to drop off at photosystem 2. And the energy from the light is actually going to help take those electrons from water and then cause water to split into oxygen and hydrogen ions. And so this is when oxygen is going to be created. The oxygen that is in our atmosphere that we're breathing in, it actually comes from these water molecules, where we're going to take those electrons and we're going to donate those electrons here at photosystem 2. And those are the electrons that are going to be excited and then leave photosystem 2. And then there's going to be more and more water molecules that come along and drop off electrons, come along, drop off electrons. And then in photosystem 2, those electrons get energized and then leave the photosystem. That's the first part here in the light reactions. And again, this is on the thylakoid membrane, the membrane that makes up those discs inside the chloroplast. So then what happens next, so that's photosystem 2, is then those electrons will go through a series of proteins that form basically an electron transport chain, a lot like the one we saw in cell respiration. And so in the electron transport chain, which I'll just go to this picture here, we're going to take these high energy electrons that got energized in photosystem 2, and we're going to pass them through these proteins, and one of those proteins is going to use the energy from those electrons to pump hydrogen ions against its concentration gradient, which is exactly what we were doing in cell respiration. In the electron transport chain, we were using the energy from those high energy electrons to pump hydrogen ions against their gradient. That's what's happening here. And so those high energy electrons, this protein right here is going to take some of that energy and then move these hydrogen ions against their gradient. If you guys look specifically at where they're headed, we're moving hydrogen ions from the stroma, which is outside the thylakoid, into the thylakoid space. So this is inside the disc. So we're moving hydrogen ions into the thylakoid space, where there's going to be a lot of hydrogen ions inside the discs. So those discs that I was pointing out, the thylakoids, inside those thylakoids, that's where we're pumping a bunch of H pluses. So there's a really high concentration of H pluses inside the thylakoid, those discs, and a low concentration outside the thylakoid in the stroma. And so that's what's happening here. So that's active transport, and he's using the energy from those electrons to do that. So by the time those electrons get passed along here, they're now low energy electrons. So their energy was taken, and now they're low energy electrons again. And then those low energy electrons are going to arrive at photosystem 1. So then they're going to be dropped off here at photosystem 1. And so then we'll go to photosystem 1 and see what happens. In photosystem 1, though the same thing is going to happen as in photosystem 2, we're going to see all this light energy be absorbed by these pigment molecules. So there's all these pigment molecules in photosystem 1. They're absorbing all this light energy. They're focusing all that light energy into these central pigment molecules. In this case, it's called P700. It's a slightly different chlorophyll. Doesn't really matter. But all that energy is being absorbed here, and it's going to be used to re-energize those electrons. So those low energy electrons that just arrived from the electron transport chain, where they lost their energy, are now going to be re-energized in photosystem 1. All this light energy is going to be used to re-excite those electrons. So now those electrons are very energetic, very high energy electrons. And once they become high energy electrons, they get booted out of this photosystem, just like they were booted out of the last photosystem when they became excited. And then this time, once they get bumped out of photosystem 1, they then are given to this last protein here, which is called NADP plus reductase. And his job is to reduce NADP plus. So there's this protein here who is going to take NADP plus and reduce it, reducing meaning gain electrons. So he's going to give these high energy electrons that are leaving photosystem 1. This protein is going to give those electrons to NADP plus, who's then going to carry those high energy electrons away as NADPH. So now those electrons are leaving as high energy electrons carried by NADPH. And so we say that NADP plus is the final acceptor of these electrons in this electron transport chain. So in photosynthesis, there's an electron transport chain, like I'm showing you right here. And these electrons are being passed along, passed along, passed along. And the final person to accept those electrons at the end of the electron transport chain is NADP+. And so that's different than in cell respiration. In cell respiration, there was also an electron transport chain, but the final acceptor of the electrons at the end of the electron transport chain in cell respiration, that it was oxygen who took the electrons at the end. In this case, it's this guy, NADP+, is going to carry those electrons away. And so that's the gist of what's going on here so far. And so just to re-summarize real quick here, you guys, in photosystem 2. So photosystem 2 is the first photosystem. This confuses students too. It's called photosystem 2, but it's the first one. And then there's photosystem 1. It's the second one. You might be wondering, like, why is that one photosystem 2 and this one's photosystem 1 if this is the first one and that's the second one? Unfortunately, over the history of science and biology, photosystem 1 was discovered first. So they discovered this one first in plants. So that's why it's called photosystem 1. And then they realized later on, shortly after, that there's actually a second photosystem, which is really the first one. But since it was discovered second, it's called photosystem 2. So be careful. Photosystem 2 is the first one. And anyway, in photosystem 2, we're taking energy from the light, and we're using that energy to excite electrons. And those electrons came from water. So water is here dropping off electrons at photosystem 2. Those electrons get excited. They leave the photosystem. They go through an electron transport chain who uses the energy from those electrons to pump hydrogen ions into the thylakoid space. Then they arrive at photosystem 1 as low energy electrons because they just got their energy taken from them. And so now they're low energy electrons. They arrive at photosystem 1. And photosystem 1, energy is again used from light to re-excite those electrons. So those electrons absorb energy from the light that's being absorbed by these pigment molecules. And then they become high energy electrons again. They leave photosystem 1 and then are ultimately given to NADP+. And now those electrons are here with NADPH. So that's the story so far. And then in this process, when water was dropping off electrons, that's where oxygen is made. So now we know why photosynthesis makes oxygen. This is it right here, you guys. This is where that oxygen comes from. Right here, it was water, and now it's oxygen. And that's going to be released by the plant. So then the only last part here to talk about is, like, why were we pumping hydrogen ions into the thylakoid space? And so that brings us to the last part, which is chemiosmosis, which should sound super familiar because this was also in cell respiration. There's a familiar protein also on this thylakoid membrane called ATP synthase. We've seen this guy before. ATP synthase is a protein that synthesizes ATP. And what it does is it uses the energy from these hydrogen ions going back down their concentration gradient. So that movement of hydrogen ions from an area that has a high concentration to an area that has a low concentration, that flow of hydrogen ions is going to power the production of ATP by this guy, ATP synthase. We call that process chemiosmosis. It's the exact same thing that happened in cell respiration. But be careful here because what's happening is, as you guys can see, here's the thylakoid, the disc thingy. And then this is inside the thylakoid, that's where the hydrogen ions were being pumped. So there's a really high concentration of hydrogen ions in the thylakoid. And then outside the thylakoid, which is the stroma, the space outside the discs, there's a low concentration. And so that's the direction in which these hydrogen ions are flowing to power the production of ATP. So at the end of all of this, we have some NADPH, who's now holding on to those high energy electrons that originally came from water. If you go back here through this long journey that these electrons went through, they're given to NADPH. Some of that energy was used to pump hydrogen ions, which is then used to power the production of ATP. So we have NADPH now. We have ATP. Those two things are made here to then be used in the Calvin cycle. So these guys, we're going to see them being used in the Calvin cycle, the next part, which is topic 3 of these notes. And then the only other thing that's made here is oxygen from the water. The splitting of water molecules during photosystem 2 results in oxygen being produced. And that oxygen is just going to be released by the plant. So the oxygen is done. The plant's going to get rid of the oxygen. But then the ATP and the NADPH that's made, that's going to move on to the Calvin cycle. And so that's the end here with chemiosmosis. And then this is a great slide to re-examine and to compare and contrast cell respiration with what's going on so far with photosynthesis. There is an electron transport chain and chemiosmosis taking place in the mitochondria, and there's also the electron transport chain and chemiosmosis taking place in the chloroplast. It's very important that you guys understand the very specific difference between both of them. And the mitochondria on this inner membrane, the inner membrane of the mitochondria, that's where the electron transport chain was in ATP synthase, and the hydrogen ions were being pumped into the intermembrane space. This is good review for this class. The hydrogen ions were being pumped into the intermembrane space, this dark gray area. That's where the hydrogen ions were being pumped to by the electron transport chain, which produced a very high concentration of H pluses in the intermembrane space, which would be a very low pH, very acidic. And then in the matrix, there's a low concentration of hydrogen ions. So in chemiosmosis, the hydrogen ions were diffusing from the intermembrane space back into the matrix, which was powering the production of ATP. Now in the chloroplast, hopefully you guys can explain and see the difference. In the chloroplast, this is located, the electron transport chain in ATP synthase, that's located on the thylakoid membrane, the membrane that makes up these discs inside the chloroplast. And those hydrogen ions in the electron transport chain are being pumped into the thylakoid space. So inside that, they're being pumped into the space inside these discs. That's where there's going to be a high concentration of hydrogen ions. And there's going to be a low concentration of hydrogen ions outside the thylakoid, which is called the stroma, this light gray area. And so in chemiosmosis, ATP synthase is going to use that flow of hydrogen ions from the thylakoid space, from inside these discs, to outside the discs and the stroma to power the production of ATP. So start trying to focus in here on the specifics of what's going on in these different processes. But that's basically it for topic 2 here. And so in topic 3, we'll talk about the Calvin cycle and what's going to happen to these NADPHs that are carrying these high energy electrons, and what's going to happen to these ATP molecules that were made here as well. And so that's it. Thank you guys. I'll see you in the last video.